I suggest that you begin by having the students make nucleotides, because usually you don't get bases and phosphates and sugars floating around individually, although you probably do some. But from my understanding, you will be more likely to find nucleotides pre-assembled waiting to be put into DNA or RNA. Of course, a nucleotide consists of a base, either A or T, C, G, or U, and a sugar. In the case of DNA, it's deoxyribose. And in my model, you can see down here is where you can tell whether it's ribose or deoxyribose. If there's no O down here, then it's deoxy, meaning minus an oxygen, no oxygen. So this is deoxyribose and a phosphate. And this is the same thing you find on ATP. There's 3 of these on the end of ATP, but here we see it used in DNA. It's a little connector. So the phosphate always attaches to this carbon on the sugar. You can see that the lines go off like this. So wherever there's a line that's running right off the edge of the piece of paper, that's where you can make a connection. You'll notice all the other lines are contained inside. So if it goes off, that's where you can connect. So imagine this is a flagpole that needs a flag, and it's a round flag. So there's only 2 lines that come off, this oxygen and this oxygen. You can't connect the other oxygens because there's no lines going off, only here and here. So in this case, it goes like this, and then this oxygen will connect actually to another sugar like that when we connect them together. Now for joining them, you have a number of options. You can use a glue stick, a little dab of glue stick will probably keep it, or a little tiny bit of white glue, or tape if you want the joints to be done immediately. Now I recommend 1/2-inch tape, which is unfortunately a little bit hard to get. I used to just buy this at our local Walmart, no problem. And now they only sell the thicker version. For a while they did too. This is the 1/2-inch, and now they sell a thicker, maybe 5/8 or 3/4 or something. I am going to order some special order online to make sure I have 1/2-inch tape, because it is really handy to have that narrower tape for a lot of craft projects. And here it's especially helpful because you can just go like this and tape this right on like that, and then you don't have a lot of tape going out over the edge. It pretty much exactly fills that space like that. So we have our phosphate and our deoxyribose, and then you can put any base onto this. Now down here, this is where another phosphate will go. So the one that's kind of opposite the phosphates is where the base is going to attach. And you can put any base on, and so if I put on, let's say, a G here like this, now the letter G is going to be right side up. But I could also put a C on. Now how am I going to do this? I have to turn it upside down to make the match like that. But that's okay. In this model, it doesn't matter whether the letter is upside down or right side up. Chemically, it's the same. It doesn't make any difference. So let's go ahead and let's just do this. Let's just put on a C, a cytosine. And then I'm going to set my nucleotide aside for a minute. I'll build another one. I need a deoxyribose, and I need a phosphate. And try to make sure that you get the lines matched up pretty exactly like that. Really try to be careful that they're matched up, because your model will go together better in the end. And then let's see. Let's put on an A, and then I will mention that little extra piece that hangs out. So the A and the G have a little extra bit like this hanging off. And that is to allow you the flexibility of either gluing them in place. You could actually glue the T on like that, or tape it. Or you can also paper clip them. So if you want to be able to unzip your DNA and simulate the making of messenger RNA, you want to be able to take this apart. So because it overlaps a little bit, you can just slide a paper clip on right there. So let's make a few more nucleotides. Now we can put some of the nucleotides together to make two rungs of DNA. So remember that C and G go together, and in this case you can't forget because look, they match, the shapes match. If you try to put a T on, it doesn't work. This has two little dots, this has three, which I've indicated by that shape. So you can see when there's a mismatch, the DNA fixer enzyme doesn't have too much difficulty realizing that something's wrong because it just doesn't fit. So let's match this up, and I'm going to tape mine. Again, you can use glue stick or white glue if you'd like to. And I'm going to match my A, T. Now I can join these rungs together right here and right here. You see there's an oxygen hanging. So this is a tricky part to get to go flat. Now you can overlap as necessary like that. If it doesn't match up perfectly, that's all right. As long as you get the correct oxygen attaching like that. Now these letters are all right side up. They look proper, but it's perfectly okay to add a rung with it upside down because of course not all T's are on this side of the ladder, not all C's. Sometimes they're flipped, it'll be TA or CG, in which case the letter in our model is going to look upside down, but remember chemicals can't be upside down, so it's just as good to go like that. And then of course that would always match up with an A. So your final model will have a mixture of right side up and upside down. Now when you go to make your RNA, there's two things you have to remember. Number one, instead of deoxyribose, we have ribose, which has an oxygen O right there. So we need the ribose sugar. I made mine a slightly different shade of purple just because I had those two purples on my shelf. You can make it any color you like. So we use that, same as deoxyribose. We put the little phosphate flag on it, same way. And then in place of T, we have U. So all the others are the same. C, G, and A stay the same, but we have to use this U instead of the T. So we can see the difference if we lay them side by side. We see on the T there's a carbon here and 3 hydrogens, and here on the U there's no carbon, there's just 1 hydrogen. So right there, that's the difference between the U and the T. And that arrangement is better for RNA, the way it has to function. It's better to not have that carbon there. So otherwise, they join up the same, they act very much the same. And of course the U will attach to an A. That's how it'll match up during transcription. So then you can join your ribose nucleotides same way you did. You use the same A and G and C that you did with the DNA. The only difference is U. And attach them. course, it doesn't have another side. RNA is one-sided. So there's your finished piece of RNA. Perhaps it's messenger RNA. One more important thing to notice about your model is that there are little numbers next to the carbons. And this is how chemists keep track of which carbon is which. Instead of saying the one on the right or the left, top or bottom, they gave them numbers. They started with this carbon and then they went around clockwise. 1, 2, 3, 4, 5. Now the main thing they use these numbers for is to keep track of which end is which on the string of nucleotides, because even though you have 1,000 or 1,000,000 nucleotides strung together, there's going to be 2 of them that are on the end. Somewhere you're going to have a last one on this end and a last one on this end. So again, instead of saying the top or the bottom, they use the carbons. They say, well, this one, the last carbon, is the 5. Number 5, this end isn't joined to another nucleotide. And what about this end? Well, it's this carbon. Number 3 is not attached to anything down here. So this is the 3' end, where the last nucleotide has an empty 3'. And here, this is the 5' end, where this end isn't attached to another nucleotide.